All right, let's dive in. We're shrinking down, way down to the molecular level for this deep dive into receptor-ligand binding. You've given me, well, us, a stack of papers and textbook chapters. I'm guessing you want, or need, I guess, a clear picture of this whole intricate dance and why these tiny interactions, well, why they matter so much, especially when we think about clinical settings.
Absolutely, yeah. It's fundamental, really. I mean, this process underpins so much, like how our bodies even respond to medicines, how cells communicate with each other. It's all connected.
Okay, so let's zoom in,
Yeah.
weigh in, set the scene. What are we actually looking at when we say receptors and ligands?
Okay, so imagine a bustling city, all right? Lots going on. Now, the receptors, those are like specialized buildings, each one with a specific function embedded right in the cell's membrane. Then we have, well, think of them as a delivery trucks, carrying those vital packages. Those are our ligands, hormones, neurotransmitters, even drugs. They're all looking for the right receptor to bind to.
So the goal is like a successful delivery, one that triggers some action inside the cell, right?
Precisely. But this isn't just some random delivery service. It all depends on this concept of, well, we call it affinity. Basically, how strongly a ligand is attracted to its receptor. So the higher the affinity, the more likely they are to, actually connect.
Okay, so affinity, it's like how much a ligand and a receptor like each other. What determines this attraction? Is it, I don't know, love at first sight? Or is it more scientific than that?
It's definitely more on the scientific side of things. Think about it like a lock and key. The size, the shape of the ligand, it needs to match the receptor's binding site. And then, well, you have charge distribution too. Opposite charges attract, so that plays a role.
So it's a combination of factors, kind of like I don't know, a molecular dating profile.
Right.
But even if they, if a ligand and receptor have great chemistry, do they just like stick together forever once they bind?
Not quite. It's a bit more complicated, I guess. You see, the binding process has these reversible steps. So the ligand approaches first, makes contact, then forms a stable complex if the fit is just right.
Oh, so it's not a one-way street. So they can actually come apart.
Yeah, absolutely. And that's actually really important. It's this like dynamic equilibrium.
Yeah.
Ligands are constantly binding and unbinding from their receptors. It's like, a busy train station, passengers coming and going, always movement.
I like that. So even when we talk about, binding, it's not static.
Mm-hmm.
There's this constant flow of molecules attaching, detaching.
Exactly. So as more ligands come in, the chances of binding increase, and eventually we reach a state of like equilibrium. That's where the rate of binding and the rate of unbinding are equal.
Equilibrium. So things are balanced, but not necessarily at a standstill.
Right. It's a dynamic balance. The energy, the movement, it all stays pretty consistent, even though individual molecules are constantly coming and going.
Okay, I think I'm following. Now let's talk about those key players on the dance floor, so to speak. So we've got the receptors, the ligands. Anyone else we should know about?
Oh, of course. Can't forget about the receptors themselves. They're not just like passive docking stations. We're talking about complex protein molecules, right? Embedded right in the cell membrane. They're like the gatekeepers, deciding what gets in and out of the cell.
Right, so like a club with a very selective bouncer.
Yeah,
Only certain molecules with the right credentials get in.
exactly. And each receptor, it has a unique binding site,
Mm-hmm.
carefully crafted to interact with its specific ligand. And then, of course, we have the ligands, all these different kinds of hormones, carrying instructions from glands far away, neurotransmitters zipping between nerve cells, and even those drugs designed to hit very specific receptors for a specific effect.
So many molecular messengers, it's incredible, all buzzing around, each with a destination and a purpose. But how does this cell actually respond? Once a ligand binds, does the receptor just, I don't know, magically open a door and let it in?
Now that's where things get really interesting. A lot of times that binding event, it's like a trigger, sets off this whole cascade of events inside the cell. And often that involves another key player. We call them G proteins.
G proteins, okay.
Yeah.
Those sound important. What do they do?
Think of them as relay runners, right? They take the message from the activated receptor and pass it on to the next guy, to those downstream effectors, the molecules that actually do the work that carry out the cell's response.
So it's a chain reaction. The ligand binds, receptor activates, G protein relays the signal, and boom, the cell does its thing.
Exactly. That's the system. It's this intricate relay that lets cells respond to such a huge range of signals, from things like regulating heart rate to, controlling mood or memory. And it all starts with that initial binding event.
This is getting complex, but I see why these tiny interactions are so important. So many processes rely on them.
Mm.
But how do scientists even measure this stuff? I mean, we're talking about, what, the molecular level? How do they even observe it?
That is a great question. Scientists have come up with some really clever techniques. One of the common methods is what we call a radioligand binding assay. They use radioactive ligands.
Radioactive, that sounds intense.
It is, but incredibly useful.
Hmm.
By measuring how much of that radioactive ligand binds to the receptor, they can actually measure the strength of that interaction. And we often represent that strength with what we call the dissociation constant, or KD.
Okay, KD, another one of those terms. So is it basically like how sticky the ligand is to its receptor?
You got it.
Yeah.
So a lower KD value, that means a tighter bond. But here's the thing, the KD value, it's not the whole story, especially when we think about a drug's effectiveness or its potential side effects.
Really? I thought a tighter bond was always good.
Often it is. But the way a ligand interacts with its receptor, it's more than just binding tightly. There are these different modes of binding, and those can affect the overall effect of a drug.
Different modes of binding. Okay, now you've got me curious. What are these different modes, and how do they affect things?
Well, we have what we call agonists, which are kind of the classic key in the lock scenario. So they activate the receptor and then trigger the cellular response. Think of like adrenaline binding to its receptor. That causes an increase in heart rate.
So the agonists are the go signal, basically. But I'm guessing there's a stop signal, too.
You bet. We call those antagonists. And basically, they block the receptor, so they prevent agonists from binding and activating it, like a broken key stuck in the lock, something like that. Beta blockers, for example, those are antagonists. They block the effects of adrenaline, so they help lower blood pressure.
Okay, so we've got activators and blockers. Anything else? The other ways ligands can interact with those receptors, it seems like there's got to be more to the story.
Oh, there is. It gets even more fascinating. There are also these things called allosteric modulators, kind of like sneaky sidekicks, so they don't bind to the main binding site, but rather to a different site on the receptor. And they can influence how the agonist, binds and activates.
So they don't activate or block directly, but they can adjust the response.
Precisely. They can enhance the agonist's effect, so make it stronger, more potent, or they can dampen the response, making it less effective.
Like they're turning the volume knob up or down on the cell's response. Okay, this is really interesting. But I bet it gets even more complex. What about those receptor dimers you mentioned? What are those all about?
Ah, yes. Receptor dimers. So sometimes you see receptors don't just hang out by themselves. They can pair up, forming these, we call them dimers, these dynamic duos.
A molecular buddy system. What's the advantage of that?
Well, dimerization, it can really change the receptor's properties. It can affect which ligands they bind to, how strongly they bind, even which signaling pathways get activated after that.
Like they unlock new abilities by teaming up? Does that affect drug development?
Absolutely. It's really important understanding receptor dimerization because we want to design drugs that target specific receptor complexes. Imagine a drug that only works when two specific receptors are paired up. That level of precision could really change how we treat diseases.
That's amazing. So many variations, different ligands, receptors, ways of interacting. It's a lot to keep track of. How do scientists even begin to, untangle this web?
It's a challenge for sure, but there are always new tools and techniques being developed. Remember those radioligand binding assays we were talking about? Those are great for measuring that binding affinity. But then we also have techniques like FORS, or first-order resonance energy transfer, and that actually lets us visualize those interactions happening in real time.
Wait, so we can actually see those molecules interacting. How does that even work?
So picture this. You've got a receptor and a ligand, and each one's tagged with a different fluorescent molecule. Now, when the ligand binds to that receptor, the two tags, they get close enough to transfer energy between them.
It's like a beacon that lights up when they bind.
Exactly. And then by measuring the intensity of that energy transfer, scientists can see some really detailed insights into how the binding happens, how fast the ligand binds, how long it stays bound, even those conformational changes in the receptor.
Amazing. It's incredible what technology can do now. But I have to admit, it's a lot. The complexity of all this, is there a risk of getting lost in the details, missing the bigger picture?
That's a good point. It's easy to focus on the mechanisms and, lose sight of why it matters. But remember, everything we learn about this, about receptor-ligand binding, it has the potential to make a real difference, to help us understand diseases better and to develop more effective therapies.
Right. It's not just about knowing the science. It's about using that knowledge to actually improve people's health.
Exactly. And I think that's a great place to, maybe wrap up our deep dive for today. We've gone through the steps of receptor-ligand binding, met the key players, talked about affinity, equilibrium, and even those more complex concepts like biased agonism, functional selectivity, and receptor dimerization.
It's been a lot, but it's been really fascinating. I definitely have a new appreciation for this whole world, this molecular world that's happening inside all of us. And I get why it's so important for medicine and disease.
And hopefully anyone listening will now look at those prescriptions, those side effects, those research papers a little differently. They'll understand that behind all of that, there's this whole world of interactions, shaping our health, influencing our lives, and we're just starting to understand it.
It's definitely given me a lot to think about. Who knows, maybe someone listening will be inspired to learn even more. Maybe even contribute to those groundbreaking discoveries, in medicine and healthcare.
That would be amazing. There's still so much to learn, but I think for now, we've covered a lot of ground.
We have. It's been a great deep dive.
It has. Thanks for having me.
Thank you. Until next time. So we're talking about more than just, how a drug gets into a cell, right? It's about the whole cascade, the signals it triggers, or even blocks. Lots of points of intervention.
Exactly. Think about drug development, for instance. Understanding a drug's binding affinity, how it binds, whether it's an agonist or antagonist or even an allosteric modulator, and what pathways it activates. It's all critical for designing safe and effective therapies.
It's like that molecular darts analogy, right? Hit the right target, the right response, no unintended consequences.
Exactly, but it's not always that simple. Sometimes you design a drug to block one receptor, but it might accidentally, even if it's weak, activate a different one,
Yeah.
and that leads to those side effects.
So off-target binding, hitting the wrong target, that explains some of those side effects we hear about.
It's one of the factors, yeah. And it shows why it's so important to really get these interactions for safer, more effective treatments. And it's not just about the drugs, it's about how diseases themselves mess with these pathways.
That's a good point. So receptor-ligand binding, it's not just about how drugs work, it's about what goes wrong when we're sick.
Absolutely. A lot of diseases, from cancer to heart disease, neurological disorders, they involve some kind of dysregulation of these interactions.
Dysregulation meaning?
It means something's off. In that delicate balance of these interactions, like in some cancers, certain receptors become overactive, and that leads to uncontrolled cell growth. Or the receptors could be, mutated or messed up somehow, and that causes a loss of that normal signaling.
So the communication system breaks down, either stuck on or goes silent when it shouldn't.
Great analogy. And when we understand these breakdowns, we can get clues for designing those targeted therapies. We can create drugs to block those overactive receptors or boost the ones that are underperforming.
Like a molecular toolkit, right?
Yeah.
Repairing or rebalancing those pathways.
Exactly. And that's where personalized medicine comes in. We can analyze someone's genes, see their individual receptor profiles, and then tailor those treatments. Better efficacy, fewer side effects.
Like tailoring a suit, you get a better fit when you have the individual's measurements. This personalized approach is really exciting. But before we get too far ahead, something's been bugging me. You said before that the Kale value, how tightly a ligand binds, it's not the whole story. What else comes into play?
You're right to ask that. Kdate is a useful measure, but it's just one piece. A drug's effectiveness also depends on, how it interacts with the receptor after it binds, the specific pathways it activates, and its overall pharmacokinetic profile.
Pharmacokinetic profile. Okay, that one sounds a little intimidating.
It's not that bad. It basically describes how the drug moves through the body, how it's absorbed, distributed, metabolized, and eliminated. All these things, they affect how much of the drug actually reaches its target, how long it stays active, its potential for side effects.
So it's about the whole journey, not just that initial binding.
Right. Imagine two drugs with the same KD value. Right. One might be metabolized and cleared out quickly. So overall, it's less effective than a drug that stays in the system longer, even if that second drug binds a little less tightly.
So it's a marathon, not a sprint.
Exactly. We're not just looking for a molecule that binds, we're looking for the right combination of binding, action, and those pharmacokinetic properties to get the best effect with the least side effects.
It's so complex. But all this talk about K-batter and drug development, I almost forgot. We've only just scratched the surface here. What about those fascinating concepts like biased agonism, functional selectivity? Can we talk about those a little more?
Absolutely. We talked about agonists and antagonists, but it's not always so black and white. Remember how binding to a receptor is like a trigger, setting off that cascade inside the cell? Well, some ligands can be incredibly selective about which events they actually trigger.
I'm all ears.
Yeah.
So how does that selectivity work? Can you give me an example?
Okay, think of it like this. You have a master key, right? Unlocks every door in the house. That's like a regular agonist, activates everything. But then you have these other keys, they only unlock certain rooms. Those are like biased agonists. They bind to the same receptor, but they only activate certain pathways, not all of them.
Two keys, same lock, but different doors in the house. Incredible. But why would a ligand only activate certain pathways?
Good question. Because receptors, they're not just simple on-off switches. They can trigger multiple effects, and sometimes those effects might be, well, opposing or even harmful.
So a biased agonist, it might activate the good pathways and avoid the ones that cause those side effects.
Exactly. And that's huge for drug development.
Yeah.
Imagine treating pain effectively without the risk of addiction, or lowering blood pressure without causing drowsiness. That's what biased agonism offers.
Wow, that would be amazing. But hold on, I bet it gets even more complicated. What about functional selectivity? How does that fit in?
Oh, it does. Functional selectivity, it takes this idea of ligand selectivity even further. So the same ligand, it can actually have different effects depending on the context.
Different effects, you mean like sometimes it's an agonist, sometimes an antagonist? How is that even possible?
It all comes down to the interplay between the ligand, the receptor, and the environment inside the cell. So a ligand might activate a receptor in one cell, but block it in another.
So it's like a chameleon ligand, changing its colors.
Perfect description. There are several factors that influence this. The specific receptor subtype, the signaling molecules that are available, even modifications to the receptor itself.
It's a delicate dance with so many variables affecting the outcome.
It really is, and this is where the most exciting research is happening. If we can understand what drives functional selectivity, then we can design drugs that are even more targeted, not just to specific receptors, but to specific cells or tissues.
So we're moving beyond just on and off switches, embracing that complexity to create more precise, more effective therapies.
Exactly. And it highlights how much more there is to learn. We're just scratching the surface. We're just beginning to see the potential of all these interactions.
You've definitely got me curious. It's clear how deep this field really is. But maybe we should, I don't know, step back for a second, revisit some of the key points. We've talked about affinity, equilibrium, the different types of ligands, KID, and now biased agonism, functional selectivity. It's like we've uncovered this whole hidden world, this, molecular level world. And the more we learn, the more questions there are.
That's science for you. Every answer just leads to more questions, deeper and deeper into the mystery of how things work. And with receptor-ligand binding, those mysteries, they're really important for understanding, well, health and disease.
We've talked about the different types of ligands, agonists, antagonists, those modulators, and those biased agonists. But are there other ways they can interact? It feels like there should be even more variations.
You're right, there are. Remember those receptor dimers we talked about? They make things even more complex.
Oh, right. The buddy system? I'm guessing those pairings, they're not random, are they? How do receptors choose who to team up with?
It's not really about choosing. It's more about, well, compatibility and the environment in the cell. So we have homodimers, where two of the same receptor type pair up, and heterodimers, where two different types get together.
Like a molecular dance party with different couples. But what's the benefit of pairing up?
Well, dimerization, it can really change how the receptor works. It can change which ligands it binds to, how strong that binding is, even which signals get activated afterwards.
So they get new abilities by teaming up. Does this affect drug development?
Absolutely. Understanding these dimers, it's really important when we're designing drugs. We want to target specific complexes, Imagine a drug that only works when two specific receptors are together. That kind of precision could revolutionize treatments.
It's amazing. So many variations, different ligands, receptors, ways of interacting. How do scientists keep track of it all?
It's definitely a challenge. But there are always new tools and techniques being developed. We talked about those radioligand and binding assays, right? Those are great for measuring binding affinity. But then there's also techniques like FRT, first resonance energy transfer. That one actually lets us see those interactions as they're happening.
Wait, so we can actually see the molecules interacting. How does that even work?
Okay, so imagine this. You've got a receptor and a ligand, each one tagged with a different fluorescent molecule. Now, when the ligand binds to the receptor, the two tags, they get close enough to transfer energy between each other.
So it's like a little light goes on when they bind.
Exactly. We can measure how strong that energy transfer is, and that gives us all sorts of information about the binding, how fast it happens, how long it lasts, even changes in the receptor's shape.
Technology's amazing. We can see things happening on such a small scale. It's like a whole other universe. But with all this complexity, I have to ask, is there a risk of, I don't know, getting lost in the details, missing the bigger picture?
That's a valid point. It's easy to get caught up in the specifics and forget why it all matters. But we have to remember, everything we learn about receptor-ligand binding, it can have real-world applications. It can help us understand diseases better and hopefully lead to new treatment.
Right. It's not just about the science itself. It's about using it to make a difference.
Exactly. And I think that's a great note to wrap things up on. We've explored the steps of receptor-ligand binding, the key players, the factors that influence things like affinity and equilibrium, and even some more complex concepts like those biased agonists and functional selectivity, and those receptor dimers.
It's been a fascinating journey. I definitely see this whole world of interactions in a new light now,
Hmm.
and how important they are for, well, for everything.
And hopefully anyone listening will now look at those prescriptions, those side effects, those research papers a little differently. They'll know that there's this whole world of interactions behind it all, shaping our health, influencing our lives, and we're really just beginning to understand it.
You've given us a lot to think about. Maybe someone listening will be inspired to learn even more. Maybe even be part of the next big discovery in medicine or healthcare.
That would be amazing. There's so much more to discover, but for now, I think we've covered a lot of ground.
We have. It's been a great deep dive. Thanks for joining me.
It has. Thanks for having me.
Until next time.